Hi everyone. Now in this part we are going to discuss about a topic called factors affecting microbial growth. Yes, we had gone through this topic of microbial growth in other part. You can have a detailed description there. You can go into it. Okay. In this part we are majorly concerned about the factors affecting the microbial growth. So let's have a quick recollect of what is a microbial growth. A microbial growth is defined as an orderly increase in cell number and cell size, or both, is going to be defined as microbial growth. This microbial growth is greatly influenced or affected by chemical or physical nature of their surroundings. For successful cultivation of microorganisms, it's not only essential to supply proper and balanced nutrients, but also necessary to maintain proper environmental conditions such as temperatures, carbon dioxide, all these things. Thus, for understanding of environmental influences on the growth of microorganisms became a mandatory in studying about them. So as bacteria shows diverse food habits, it also exhibits diverse response to environmental conditions. So here the growth rate of bacteria is generally influenced, as I told you, by various factors of intrinsic as well as extrinsic. In addition to the type of species and the medium, some physical and chemical factors which are influencing the growth of bacteria are going to be of like water, temperatures, aeration, radiation, pressure, pH, etc. Okay, and these are all things we are going to discuss in detail one by one now. Now coming to the first one is water, obviously. So water is one of the most essential requirement for life, and this is an important factor for the growth of the microorganisms, where 80% of the bacterial cell consists of water. And we know that obviously there are two types of the waters. One is the fresh water, which is available in the form of ponds, lakes, wells, all these things. And the second type is the salt water, that is sea water or marine water. The bacteria which grow in seawater are called as halophiles or halobacteria. Then what are these halophiles? As I told you, the one which is going to grow in the marine water, seawater, or in high salt concentration. What will be there in the seawater? Obviously, will have the high salt concentration. So, a bacteria which is going to grow well in high salt concentrations are called as halophiles. And one more term we have to remember is here is halotolerant. Tolerant means it do not require the salt, but it is able to survive at that high salt concentrations also. Those are called as halotolerant. Okay, and these halophiles are going to be classified into three types: slight halophiles, moderate halophiles, extreme halophiles. So depending upon the salt concentration at which they are going, so we are having like this: slight, moderate, and extreme. Okay, and thus water availability becomes most important factor for the growth of microorganisms. So as I told you, the availability of water depends on two factors: the water content of the surrounding environment. So up to now whatever I told you is the surrounding environment, and the concentration of the solutes dissolved in the water. So where are these solutes? That is salt, sugars, etc., that are present in the cytoplasm. So that also plays a factor in the microbial growth. So both the availability of the water depends upon the two factors. One is the water content present in the surrounding, and the concentration of the solute dissolved in the water. So these two factors, that is two types of the waters, are the majorly going to have the impact of water in the microbial growth. Then coming to the second type is temperature. So all forms of life are greatly influenced by temperature. That we know very well. For example, take us only. We cannot neither be of very safe in too low temperatures or too high temperatures. So we require a moderate. So in the same manner, certain microorganisms are also going to have the effect with their growth with the effect of temperature. So that means very sensitive to the temperatures, whereas with that of environment, which are called as poikilotherms. And the temperature influences the rate of chemical reactions and protein structure. So about the protein structure, you can go through the video that I already did. okay, where I explained about the primary, tertiary, secondary and quaternary type of the structure and how they are forming everything. You can have a look after it. Thus affecting rates of enzymatic activity. So we know that if we want to have a proper chemical reactions, we require the certain temperatures. So those temperatures are mainly involved affecting the enzymes activity in the metabolic process. Then coming to the temperature relationship of microorganisms is usually described by three cardinal temperatures. So any temperature that is going to affect an organism is majorly classified into three types. So what are those three types? They are minimum, maximum and optimum. Minimum, optimum, maximum, any order it can. So cardinal temperatures are going to be of having a certain importance in studying any type of effect of temperature as minimum, optimum and maximum. So let's see what are these things. So if you look at this picture, minimum means the lowest temperature which microorganism is able to grow. Just it is not going to have a full pleasured growth, but just it will survive. So that is going to be the minimum, and maximum is also the same. So that means beyond these limits of minimum and maximum, the bacteria will die, or it will die. But coming to the optimum is a condition of temperature at which they are going to show their maximum growth. Okay, let's see that one. So first coming to the minimum, as I told you, the lowest temperature at which organisms is going to survive, and here you can see the transport process so slow that growth cannot occur. Just it will tries to survive itself with that temperatures, that's all. Then coming to the next second one, optimum temperature, obviously I told you you are going to have a most rapid growth because of the multiplication, and that multiplication of cells is due to the enzymatic reaction occurring at increasingly rapid rates. And you are going to see the maximal possibility rate at this optimum conditions. And then coming to the maximum, the highest temperature at which growth occurs, but slightly above this point, kills microorganisms either by denaturation or denaturating the enzymes or the proteins, or by the destruction of the cell membrane. So that's how the maximum temperature, that's the end point beyond which it will literally die. So that's how for any organism these three are going to be minimum tolerant, maximum tolerant. That means beyond that they are going to die. So on the basis of a temperature relationship, bacteria are going to be classified into different types. So let's see what are they. So on the basis of temperature relationship, as I told you, we are going to have the different groups, that is psychrophiles, mesophiles, thermophiles, hypothermophiles. Psychrophiles, obviously psychro means cold, and then mesophiles means middle, that means a normal temperature that we are surviving. And then thermophiles, high temperatures, and then hypothermophiles is going to be of extreme temperatures at which they are going to survive. So that if you are going to look at this picture, so here is the psychrophile. Okay, so they can grow at about 0 degrees, and they grow best at a temperature of 15 to 20 degrees. So in between that it is going to be grown. Okay, and the best examples are going to be flavobacterium, and the cell membranes of psychrophiles consist of unsaturated fatty acids and remains semi-fluid when cold. So the enzyme system functions well at low temperatures. So that's the main reason why they are able to survive, because of having cell membranes, a unsaturated fatty acid, and making the internal components to be of in the semi-fluid when the temperature is very low. Then coming to the second type is mesophiles. These mesophiles grow best at 20 to your 49 or 45 degrees centigrade. Here it is 45. Okay, and mainly all the organisms, main organisms or microorganisms, fall in this category of mesophiles. The best examples, all human pathogens, as they grow best at 37 degrees centigrade. Example is E. coli. And then coming to the third type is thermophiles. Thermo means heat. Okay, they grow over a range of 40 to 75 degrees centigrade or 70 degrees centigrade, or obviously 50 to 70 degrees also. These microorganisms possess a more heat stable enzymes, like how this is going to have a special membrane consisting of fatty acids in the psychrophiles. In the same manner, the thermophiles are also going to have some heat stable enzymes which are going to perform the protein synthesis. Okay, and not only that, the cell membrane also has saturated, here it's unsaturated, here they are having the saturated lipids with high melting point. So obviously several algae, fungal and protozoan species have temperature maximum at 55 degrees centigrade, and we are also having the Dermis aquaticus and Aspergillus fungigatus or the examples of this kind of thermophile, and here is a Bacillus thermophilus is another example, here it was. Okay, so this is all about the thermophile, where they are going to tolerate the high temperatures of ranging from 40 to 75 degrees or 70 degrees centigrade. Then coming to the next one is the hypothermophiles. So here some bacteria, especially archaebacteria, can grow at 100 degrees or even high. Okay, so those are going to be of best example. Thermococcus and Pyrolobus fumari are going to be the best examples of where they can grow at high temperatures. Okay, so this is all about the different types of classification of the bacteria depending upon the temperature at which they are growing. One is a psychrophile, low temperatures; mesophile, the ordinary temperatures of ranging from 20 to 45 degrees. Then coming to the thermophile, high temperatures from 50 to 70 or for 45 to 75 or 70 degrees. Then hypothermophiles beyond 75, that is 80, and some may be of even 100 degrees or more. So then we call them as hyperthermophiles. So this is the second factor that we have discussed, is temperature. Then moving to the next one is aeration. Okay, so here just a little bit I want to tell you about this one is, each microbe shows characteristic temperature depending upon and possess its cardinal temperature. That's what I told you, the cardinal temperature: minimum, optimum and maximum growth. Okay, so I have discussed about in detail about all those things, and then we'll move to the next one, aeration. So here in the aeration, so what you are going to discuss about aeration, oxygen requirement plays an important role in the growth and growth rate of microorganisms. And these microorganisms vary in oxygen requirement. So these can be of either aerobes or anaerobes. In the aerobes, we are going to have the obligate aerobes and as well as the microaerophils. So let's see into the picture, you are able to see this. So here I told you the aeration, depending upon the oxygen requirement, the microorganisms are classified into different types, aerobes and anaerobes. So aerobes, the one which require the oxygen for their survival or for their growth, is going to be called as aerobes, and in which again we have the classification called as obligate aerobes and microaerophils. So obligate aerobes, here you can see they require the oxygen for their survival. So if you see in the test tube, here the microbes are present only on the top layer. That means where the availability of the oxygen is there only they are able to survive. So that's why we call them as obligate aerobes, that they should require the oxygen. It is must, like us. If you are not going to breathe the oxygen, you will die. In the same manner, these organisms also require the oxygen, otherwise they won't grow. So the second type is the microaerophils, the classification of the aerobes, where they require only low concentration of the oxygen. So that's why you see some sort of a sub layer of availability of the oxygen, where the oxygen is very low level. Okay, so that's why these are called as microaerophils. So that's how we have classified microorganisms into two types depending upon the aeration, what is aerobe, and second one is the anaerobe. And in that also, aerobes are going to be again of two types depending upon the utilization of the oxygen, that is the free oxygen, and the second one is the low concentration of the oxygen. And here are the examples of obligate or strict aerobes, that is Staphylococcus, Pseudomonas and Bacilli. and here microaerophils comes under the streptococcus or campylobacter. And then coming to the second type of classification depending upon the aeration of oxygen requirement, that is anaerobes. So here in the anaerobes, we are having the three types. So the one which do not require the oxygen or being called as anaerobes, and if that is must, that called as obligate or strict anaerobes. So here complete down you are able to see the growth of the organisms. And there are two more types in the anaerobes. One is the facultative anaerobes and second one is the aerotolerant. Facultative, they grow both in the presence or absence of free oxygen, or these do not require oxygen for their growth but grow well in the presence. So here you are able to see they will grow in the presence of the oxygen, and they are going to show their maximum growth in the presence of oxygen, but at the same time in the absence also they are able to grow. Then we are going to call them as facultative. The examples are Vibrio carva species. And then coming to the aerotolerance, they do not utilize the oxygen but can grow or survive in its presence. Facultative is able to grow and utilize it in presence or absence. But here aerotolerance or a kind of anaerobes which do not utilize the oxygen but they can tolerate. The name itself is saying that they can tolerate the presence of oxygen in the medium or in the environment. So the best example are lactobacilli, anaerobic streptococcus and enterococcus faecalis. So that's how, depending upon the aeration or oxygen requirement, the microorganisms are majorly classified into aerobes and anaerobes. And these aerobes are again of two types, that is obligate aerobes and microaerophils. And then coming to anaerobes, we are having the three types, that is obligate anaerobes, then facultative anaerobes, then aerotolerant anaerobes. So this is the third type of factor that is influencing the microbial growth. And then moving to the radiation, here one more thing I just want to mention about the mechanism behind this aeration, which is very important. Maybe some of the students are not interested in learning, but this is for those who are interested what is happening, why they are being allowed to grow in more aeric condition and not in an anaerobic condition. Let's have what is happening, why this anaerobic bacteria is dying in presence of oxygen, why these people bacteria are able to grow in the presence of oxygen. Means these varying relationship between the microbes and the oxygen appear due to different factors such as protein inactivation and the effect of toxic oxygen derivatives. Okay, so bacterial enzymes can be inactivated when they interact with the oxygen. Example, take the nitrogenase enzyme. That nitrogenase enzyme is a useful, you all know that in the nitrogen fixing enzyme, where in the normal condition, that is in the presence of oxygen, the nitrogen fixation do not occur, isn't it? So that's why they are going to be kept they're going to be in the form of root nodules, where they are going to provide the conditions of anaerobic, where the nitrogen enzyme is going to be of very active in condition, isn't it? So if oxygen is going to be there, that enzyme is going to be inactivated. So because this nitrogen enzyme is sensitive to oxygen, as I told you, and represents a good example of interaction between the enzyme and the oxygen. So during metabolic process of even your flavoprotein, your electron transport chain, the flavoprotein reduces oxygen to form some sort of compounds like hydrogen peroxide, superoxide radicals, and then some hydroxyl radicals, OH minus. Okay, so these compounds are highly toxic and being powerful oxidizing agents and can cause the destruction of cellular molecules. So because of all these reasons, these anaerobes cannot survive. Then how a bacteria of aerobic is able to survive by avoiding all these things? So in order to survive, bacteria must be able to protect itself from oxidizing agents, isn't it? So all aerobes and facultative anaerobes is going to possess two main enzymes, namely called as SDS, SOD, that is superoxide dismutase enzyme, and one more is the catalase enzyme. So these two, that is superoxide dismutase and catalase enzymes, are very playing a crucial role in protecting the aerobes. So and deleting the lethal effects of these oxygen products. So what are the oxygen products? That is hydrogen peroxide, then super radical, that is O2 minus, and then hydroxyl radical, OH minus. So the oxidizing property of this superoxide is nullified by superoxide dismutase. That's the enzyme, and it is converted into the oxygen and hydrogen peroxide. And the enzyme catalase decomposes the hydrogen peroxide into oxygen and water. So now the aerotolerant bacteria like lactic acid bacteria, so these things possess these enzymes, so obviously they are able to survive by removing the oxygen, and they are going to have the normal growth, where the strict or obligate anaerobes do not possess these enzymes as protective agents, and that's how they can grow in the presence of oxygen. Since all obligate anaerobes lack these enzymes, obviously, so they are not going to have any growth or low concentration, and therefore susceptible to oxygen, and they will die. So this is the actual mechanism that is happening in the anaerobes, where they cannot tolerate, and how the aerobes are going to protect it, because of having the two main enzymes. One is SOD, that is super oxide dimutase, dismutase enzyme, and second one is the catalase enzyme. Okay, so let's move to the next type of uh, factor which is influencing the growth, that is radiation. Some electromagnetic radiations, particularly the ionizing radiations, example are X-rays, gamma rays, are very harmful to microbial growth. That's the reason why we are using these radiations in the sterilization purpose. And low levels of these radiations may also cause mutations and may directly result in the death of microbes. And the high levels may directly cause the death of microbes. So some may be involved in the indirectly manner when low levels, and high levels directly kill the microbes. This is all the things of X-rays or the gamma rays. Then coming to the how it is going to break, that means how these ionizing radiations are involved in killing the microorganisms or harming the microorganisms, is they're going to destroy the ring structure or breaks the hydrogen bonds of the double bonds and polymerize just the certain molecules. So we know that there are going to be of hydrogen bonds between the DNA strands, and if they are all going to broke down, and not only that, these phosphodiester bonds are also being broke down, where it is going to get denature of this DNA structure, and obviously it lost its nature and the bacteria will die. And then coming to the non-ionizing radiation, example are going to be the UV rays, ultraviolet radiation is going to be lethal to all types of microorganisms, and the UV radiation of about 260 nanometers is going to be considered as most lethal radiation. So then how it is going to impact? The X-rays are going to break the double bonds in between the strands, and that's how they are going to affect. Then how these UV radiations are going to cause what we call lethal effect to microbes means, we know that you're going to have the bonding between the nitrogen bases of the concerned complementary bases in a strand. Now what these UV rays are going to do is, the UV rays are going to primarily form some sort of thymine dimers. That means adjacent thymine dimers. Here you can see. So actually the bonding should be in between the two complementary strands. But here what is happening? There is going to be of adjacent thymine dimers are formed in DNA to cause the damage. So if once this kind of the things are formed, so obviously that was a damaged DNA. And now in this damaged DNA, the thymine diamonds, that is, they are going to be covalently and attached covalently with one another adjacently, and obviously they are going to interrupt the DNA replication. And if the DNA replication is interrupted, obviously that is leading to the interruption in the function of the concerned metabolism, and that's how the bacteria is going to have the inhibits of its growth and causing the lethal to all microorganisms. Okay, and along with this radiations, in some bacteria, that is photosynthetic bacteria, the excited photosensitizers transfer the energy to oxygen. That oxygen is going to be resulted in the singlet oxygen, which is a dangerous one. Okay, because it's very that singlet oxygen is very reactive and powerful oxidizing agent and quickly destroys a cell. So it's nothing but a microbial cell. So probably this singlet auxin is a main weapon employed by our phagocytes to destroy the engulfed bacteria. Okay, so that's an advantage for us, but here bacteria will die. Then coming to the next type of the factor that is influencing is a pressure. So pressure, yes, pressure. So we can't bear much pressure. So in the same manner, any cell can't bear much pressure. So normal life of microorganisms on land or in the water surface or on the water surface is always subjected to a pressure of 1 atmosphere. Yes, 1 atmospheric pressure. Some microorganisms even survive and grow more rapidly at higher pressures, which are being called as barophiles. The examples of the barophiles are going to be Protobacterium, Colvilla, and Shivanida. So these barophiles is a bacteria. Barophile is a bacterium that I told you, which prefer to grow or exclusively grows at moderately high hydrostatic pressures, such as a challenger deep in the marine trench of about having a depth, so that means inside is of 10,994 meters. At that condition also you are able to see the growth of these microorganisms. So such type of the bacteria is being called as a, so it's not basophile, sorry, it's a barophile. Okay, so barophile. Then coming to the next type of the factor that is very important, though it is last, so it's very important. So last, though it is last, it's not least. Okay, pH is also another important factor that it is going to have a major impact on the growth of microbial growth or microbes. So here, what is a pH? So it is a negative logarithm of hydrogen ion concentration. The pH scale ranges from 1 point, that is 1 to 14. We know that what is a pH scale. So this is a pH scale where you are able to see from 1 to 14, okay, with a neutral point of 7. So pH below 7 is going to be considered as acidic in nature, and the pH above 7 is going to be considered as basic in nature. So that's how the 7 is going to be the neutral, and below of it is going to be the acidic in conditions, and above is going to be called as basic or alkaline in conditions. Like temperature, every microorganism is going to have an optimum pH range over which it grows. The basic tolerance range of pH for their growth are going to be differed or going to be classified into different types. So what are those different types? One is going to be called as acidophiles, the bacteria which prefer to grow in the acidic pH is going to be called as acidophile, and the bacteria which prefer to grow at uh, alkaline conditions are being called as alkalophiles or basophiles, and the one which prefers to grow at the neutral pH obviously called as neutrophiles. So here comes the three types of the bacterial groups depending upon the pH. One is the acidophiles, organism which require pH range of 5 or less for maximum its growth is going to be usually called as acidophiles, and the examples of thiobacillus thioxidans where the pH is 3 and the microfungi is 4 to 6. Then coming to the alkalophiles or basophiles I told you, so the one which is going to grow above 8.5 to 11.5 with the optimum pH growth is 9.5. Example is alkaligens. Fascicuti is a bacteria which is going to grow at the pH of 10.5 here. And then coming to the neutrophils, I told you where they are going to have a pH range of 5.5 to 8, not 5.5, it is 6.5 to 8. Most of the pathogenic microorganisms are going to have the pH range of this one. Okay, then here is the thing. So at the, if maximum of the microorganisms are going to show them may optimum growth, that means where they are going to have the more growth is going to be considered as the optimum. So there we have seen in the cardinal temperatures minimum, optimum and maximum. So here at the optimum pH we can see the maximum growth of the microorganism. So this optimum pH is going to be different for each type of the microorganism where we have classified them as acidophils, neutrophils and alkalophiles. So the acidophiles, as I told you, they are going to have a range of from 1 to 5.5. Then neutrophils is going to 5.5 to 8.5 or 8, and the alkalophiles may range from 7.5 to 11.5. So this is how the pH is going to have its impact in the growth of microbials or microbes. Okay, so that's we finish the major important factors which are affecting the microbial growth. So what are those 5 or 6 factors that we have discussed are water, temperature, aeration, radiation, pressure, and pH. So water we have discussed, temperature we have seen the 4 types of bacteria, that is we are going to have psychrophiles, mesophiles, dermophiles, hypodermophiles. Then we had gone with the next one is your aeration. We have seen the aerobes, isn't it? So temperature for aeration we are going to have how many? 2 types majorly, that is aerobes and anaerobes. Then we have gone with the radiation, ionizing radiation effect and the non-ionizing radiation effect. Then pressure, barophiles we have discussed, and the pH, the last one that we have discussed is estophiles, basophiles and neutrophiles. So that's how we end this factors affecting microbial growth topic. Thank you.